Bismillahir Rahmanir Rahim. Now, guys, it's chapter number 5, membrane potentials and action potential. In the first paragraph, they have discussed that the electrical potential is present to the outside of every cells, but some cell uses this electrical potential is a basic for performing their job, and some cell produce changes in their electrical distribution in order to carry out some important functions. So this was simple introduction. Now the basics physics of membrane potential. Membrane potential caused by ion concentration differences across a selectively permeable membrane. Now if we take a look through the figure 5.1, for example, this assume that the membrane is now only permeable to the potassium. So the potassium, the potassium is more on the inside of the cell as compared to the outside. So the potassium has the natural capability and has the natural ability to flow from the high concentration to the low concentration. So in this way, the potassium will diffuse through the potassium channel to the outside of the cell and will create and will create negativity because of the potassium is a positive charge and it carrying the positive charges from the inside of the cell membrane to the outside and creating the negativity inside. What causes another factor which contribute to the electronegativity, these are the proteins, sulfates, etc. These substances are entrapped inside the cytoplasm which cannot exit the cell. So because of these negatively charged proteins and amino acids, etc., these remains inside the cell and the positivity charge are exiting the cells. So because of this, the negativity is developed inside the cell membrane. Now a point will reach now the potassium will diffusing outside continuously, and a point will reach that the potassium will not be able to diffuse furthermore. That point is, this is the diffusion potential of the potassium, and this is minus 94 millivolts. This is the voltage of the potassium at which the potassium ion cannot further diffuse from the inside to the outside. Now assume that the membrane is now permeable only to the sodium ion. And the sodium ion is more on the outside as compared to the inside. So the sodium ion has also the natural capability to diffuse inside the cell. So the sodium will diffusing and will carrying positive charges to the inside of the cell and will create positivity inside. And in this way a point will reach that the sodium will also stop diffusing. This point is plus 60 millivolts, plus 61 millivolts. This 61 millivolts is the diffusion potential of sodium, and which the sodium cannot further diffuse through the membrane. So this was a simple discussion for potassium and sodium. The diffusion potential for the potassium is minus 94 millivolts at which it stop diffusing, and for sodium it is plus 61 millivolts. It also stop diffusing at this voltage. Now this was about the membrane potential caused by ion concentration differences across a selectively permeable membrane. Now we will discuss the Nernst equation describe the relationship of diffusion potential to the ion concentration difference across a membrane. What is the relationship between the diffusion potential and the concentration differences across the membrane? For example, first of all, what is the Nernst potential? The potential at which the diffusion of ion cannot occur further, this is called Nernst potential. For example, if this is the cell membrane and these are the ions which are more on this side and less on this side. So these ions will diffuse again and again, these will diffuse, and when it reaches to a point of potential that it cannot diffuse further, this is called Nernst potential. So this is the relationship between the concentration difference across the cell membrane and the Nernst potential. That initially the concentration was high on the outside and was less on the inside, and then it start diffusing to the inside. A point will reach that further diffusion will not occur. So this is called the Nernst potential. The potential at which the ions stop or movement of ions or diffusing of ions stop across a barrier. The potential at which the ions stop or movement or diffusing stop across a barrier, their potential is called their potential is called Nernst potential. Nernst potential. So this was about the Nernst potential and the relationship between the Nernst potential and the concentration difference. Now the formula of Nernst potential. This is the formula of the Nernst potential. This is Z is the charge for which the Nernst potential has to be calculated. Now using this formula, two points should be noted. One is when using this formula, the outside extracellular fluid potential will be taken 0. The calculated potential will be on the inside. So these 2 points should be noted. Another point is that if the charge which is coming to the outside of the cell is positive, so the sign of the Nernst potential will be taken negative, and vice versa. If for example, if if the concentration of the charges on the inside is 10 times to the outside, then the log 10, if we take the log of 10, the log of 10 is 0. So the log of 10 is 1, sorry. So the Nernst potential calculated to be minus 61 millivolts inside the membrane. This was about the Nernst potential. Now the Goldman equation is used to calculate the diffusion potential when the membrane is permeable to several different ions. In case of one ion, we can calculate the Nernst potential or diffusion potential through this formula. But if the membrane is permeable to more than one ion, then how can we calculate the membrane potential? We can calculate this through the help of Goldman equation. Goldman equation, another point should be noted in the Goldman equation, that the if the charge is positive, then we will take the concentration will be divided, the inside concentration will be divided by the outside. But if the charge is negative, then the outside concentration will be divided by the inside. So this is the difference. When this is positive, the inside concentration will be divided by the outside. When the charge is negative, the outside will be divided by the negative. The Goldman equation depends on 3 factors. First of all, using the Goldman equation and calculating the membrane potential, it depends on 3 factors. First of all, it depends on the polarity of charge. It is positive or negative or neutral. Depends on the permeability, that how much the membrane is permeable to a particular ion. And the third is the concentration of a substance. How much concentration of a substance is present on the respective site. So these are the 3 factors which can affect the membrane potential. Now, what is the importance of the Goldman equation? The importance of the Goldman equation is 4 folds. That first of all, with the help of Goldman equation, we have determined that sodium, potassium and chloride are the most important ion in developing the action potential in the neurons. Second, we have also determined that how much a particular ion has contributed to the potential. We can also calculate it from this formula. Third point is, when there is a gradient from one side to the other side of a particular charge, then what charge will be developed on that particular side. Fourth point is, we have determined that fourth point is, we have also determined that the changes in the neuron membrane in the permeability, which is changing for the sodium and potassium, is rapid as compared to the chloride. So the sodium and potassium are the most important ion in generating the action potential. So these were the 4 main points which has which has been proved from the Goldman equation. First, which ions are important for the action potential. Second, contribution to the voltage, which ion contribute to the voltage. Third, the gradient. Fourth, which ions are important for the generation of action potential across the cell membrane. Now, the resting membrane potential. So, this what is the resting membrane potential? It is actually a refractive period between the two consecutive action potentials. So, when one action potential has developed, then this action potential must be terminated for the generation of another action potential. So there will be some refractive period. This refractive period is called the resting membrane potential. Now this table has shown different types of action potential, that the neurons -60 to -70, skeletal muscle -85 to -95, smooth muscle -50 to -60, cardiac -80 to -90, hair -15 to -40, astrocytes -80 to -90, erythrocyte -8 to -12, photoreceptor at dark -40, at light -70. So these are the resting membrane potential. It should be remembered. Resting membrane potential. Electrochemical driving force. What it means by the electrochemical driving force. Now I will explain this. Electrochemical driving force. Electrochemical driving force. For example, cell membrane, and this is the calcium ion, sodium ion, and potassium. The sodium ion has contributed 10 units to a particular potential. It has contributed 40 units, and it has contributed 50 units. Let's suppose these ions has for a particular potential, the contribution of these ions are 10, 40, and 50 units. For example, if we take only the example of sodium, that sodium ion, the equilibrium potential for sodium ion is let's suppose the equilibrium for sodium ion is, let's suppose it is 20. So the equilibrium potential means that the ion will stop moving across the membrane and it will be equalized on the both sides. So this is the equilibrium potential for the sodium ion. For example, if the equilibrium potential for the sodium is 20, the membrane potential at which the sodium has contributed how much of the membrane potential? For example, 30. So this is the membrane potential of sodium, membrane potential. Now how can we calculate the electrochemical driving force for the sodium? We will calculate, for example, we will minus 30 from the membrane potential, we will minus the equilibrium potential minus 20. So it comes how much units? 10 unit. So if the membrane potential is 30, if the membrane potential is 25, and the sodium potential also, and the equilibrium potential for sodium also becomes minus 25, so the driving force, this is the driving force, will be equal to 0, and there is the driving force is equal to 10. So this is the electrochemical driving force. This was for only the sodium, that for example, this is a particular potential at to which the calcium, sodium, and potassium has contributed its respective potentials. And if we take one ion sodium, the equilibrium potential for the sodium is, let's suppose, 20, and the membrane potential for sodium is 30. The equilibrium potential is a potential at which the sodium equalizes its moment across this membrane. Equalize its moment, that it cannot move, it cannot diffuse, so it equalizes its respective moment across the membrane. This is the equilibrium potential for the sodium, and it is the membrane potential for the sodium. It can be any potential. So when we divide when we minus the equilibrium potential from the membrane potential, we will get the driving force. In this case, for example, if the membrane potential is if the membrane potential for the sodium is 30, and the equilibrium potential is 20. So we will get the driving force of 10. And this driving force, the sodium will utilize this driving force to move its ion across the membrane. But if this membrane potential becomes equal to the equilibrium potentials, then both will neutralize each other and the driving force will be zero. So we can also, this was the example for the sodium. This can also be applied for the potassium, for the calcium. So this was electrochemical driving force. That what is electrochemical driving force? Now measuring the membrane potential. How can we measure the membrane potential? The membrane potential can be measured by this apparatus. It consists of micropipette. This micropipette has been filled with an electrolyte, which is potassium chloride, and this micropipette is traverses across the cell membrane and is present inside the cell. And this is the indifferent electrode which is attached to the negative pole, and the micropipette is attached to the positive pole, or this is the voltmeter. So these are the respective poles through which the micropipette and indifferent electrode is attached. This voltmeter is highly sophisticated. Despite such a huge resistance, for example, this micropipette, the tip of the micropipette is very much small, so it will face a huge resistance. And despite this huge resistance, this micropipette can calculate slight changes across the cell membrane. So using this apparatus, we can calculate the changes in the electrical potential across a particular membrane. For example, if this apparatus is moved to an area where the potential is decreasing from from 0 to minus 70 millivolts, it will decrease. It will showing decreasing voltage. And if it passes through an area of rising voltage, then the voltmeter will show the voltage is rising. So we can calculate the changes across the cell membrane through this apparatus with high correct value.